My name is Najib ElSayed, and I'm an associate professor in the Department of Cell Biology and Molecular Genetics with a joint appointment in the Center for Bioinformatics and Computational Biology. My lab focuses on the study of host-pathogen interactions using genomic and bioinformatic tools. We aim to understand how pathogens, and more specifically parasites, establish infection within the human host. They do so by evading the immune response and subverting various molecular processes and modulating a variety of molecular functions in the human host. We also would like to study how the human is responding to the infection by the parasitic organism.
I'm Cecilia Fernandez. I work at University of Maryland in the lab of Dr. Najib ElSayed, and I'm a postdoc here. My work focuses on studying the interaction of Leishmania parasite with its human host. Leishmania is an intracellular parasite, meaning that it has to invade a cell in order to replicate and survive. Leishmania infects a very specific white blood cell of our immune system called macrophage, which are part of our body's first line of defense against invading microorganisms. Normally, when a macrophage ingests a pathogen, being it a parasite or a microbe, it traps it inside a compartment called phagolysosome, which will eventually degrade it using enzymes and toxic peroxides. Leishmania, the parasite that we study in this lab, however, has developed mechanisms to survive within this harsh environment of the phagolysosome. Fortunately, we're able to mimic this infection here in the lab, and using the most advanced genetic sequencing techniques, we are trying to understand which genes are turned on and which genes are turned off during this infection process. Essentially, we're trying to understand how Leishmania is manipulating the macrophage's response in order to survive within its human host.
My name is Keith Hewitt. I'm a graduate student in Najib ElSayed's lab in the BiSci program. I work on network analysis in the context of genome sciences. It turns out when you look at how genes are turned on and off in the pathogen and the host during infection, that the response on both the pathogen and the host side is very complex. It's complex because it involves networks of thousands of underlying genes interacting with one another and changing across time, driving the success of the pathogen as it invades the cell and also the response of the host. While traditional approaches have focused on the individual genes, we're trying to interrogate things at the genome-wide scale and look at all 10,000 pathogen genes and all 20,000 host genes and look for the important interactions between the two. We want to see when the pathogen is secreting proteins to enable it to invade the cell or evade the immune system of the host. What is it doing? Which proteins are acting in concert to allow this? And trying to see what are the signatures of invasion of the human cell? What is the parasite doing that allows it to invade and then survive within the cell? And what is the host doing to respond to that? And we're trying to find the kind of core interactions between the parasite and the host and look for things that are also unique to the parasite, which might be potential drug targets in the future.
My name is Eva Morgan, and I'm an undergraduate senior working in Dr. ElSayed's lab. Leishmania and related organisms are very unique in many ways. Among them, they are different from us in the way they regulate the expression of their genetic code, and we know relatively little about it. In my undergraduate thesis project, I used both wet lab and bioinformatics to understand how these parasites fine-tune the production of protein at the RNA translation level. By comparing both the infective and non-infective form, we can understand how these organisms are able to cause disease.
Our ultimate goal is to identify targets in the pathogen that will allow us to destroy it without impacting the human host.